So you've all turned in your assignment one and you got grades for it. Some of you chose to take advantage of the resubmission and submitted and address some of um, the feedback that you were given. We'll have them, I think, graded in the next few days. Those are quick. And then you'll get your new grades, which will replace your whatever your first submission was. Next week, we have the second exam. We'll have a study session later this week. I don't know if you talked about a specific time. Did you guys decide on a time? Or?
Thursday 2 to 4.
Okay, Thursday 2 to 4. You've done this once, so you know how it goes. It's just I'm gonna share a study guide, and then this study guide will be a list of short essay questions. A subset of those will appear on the exam. And then there will also be multiple choice questions that could come anywhere from the slides or the lectures. Yeah, just do a study before going to the study session. You'll get more out of it that way. And then you can, use like come with your questions so you could really understand the material. And if you miss it, there will be a recording you could watch to catch up. Any questions about the exam? The exam is not cumulative, so it's a material that we covered since the first exam. So the last 3 weeks and today, so 4 weeks of material on vision and different brain areas involved in visual processing. All good? All right, so I think we left off here last week, where we were talking about these two main visual pathways in the cortex. We talked a lot about different brain areas that were involved in taking the visual signal from the retina to the LGN to primary visual areas and going toward the parietal cortex and going toward the temporal area. And you know each of these pathways by now, like one of them is the ventral pathway that goes to the temporal lobe from occipital, and the dorsal pathway goes to the parietal cortex from the occipital. We left off talking about the inferior temporal or inferotemporal cortex. So as we looked at the selectivity of earlier visual areas, the units in earlier visual areas, we saw that they're selective to orientation of the bar or direction of motion. As we go up the temporal path, as we go up the ventral pathway, we see units that are much more selective to more complex patterns. For example, people have found out units that are selective to faces. And it really depends on the quality of face too. Maybe they respond less strongly. These are action potential. This is a raster plot. You've seen them. They respond less strongly to a drawing than to a face that is missing certain features than to a full face. So we're saying that these cells are selective for faces, meaning that they would respond most strongly when they have a face in their receptor. A lot of these face selective cells in human brain are found in FFA or fusiform face area. In the monkeys, there are multiple patches that are face selective for different orientation of faces. In the fMRI, you could detect fusiform face area fairly easily. So damage to the IT can cause damage in visual form perception. So we talked about visual form agnosia, a form of a very extreme version of this was, we saw it earlier, which was apperceptive agnosia, where you cannot really even copy an image and you cannot recognize it. But there is another form of agnosia, which is not as much, as bad. This is associative agnosia, where you could actually draw something that you're looking at, but you're not able to name the object. So it's really a matter of associating those visual features to an identity or a category. So there are different degrees of agnosia depending on the location and the extent of the damage. But in general, there is a lot of evidence that this part of the brain in the temporal lobe is involved in visual form processing. You're processing what the form of an object is, what the features of an object are, and you're associating those with a category. So a very special case of visual form agnosia is called prosopagnosia. This is when people have difficulty recognizing faces. So this is a specifically damage to the face processing area, and some people are not able to recognize faces, but they could use other features. They could use the way people talk, their way of walking, their gait, their smell. They could use other features of the person to recognize who the person is, but they cannot really use the features of their face to associate that to their identity. So they're not able to process face features. And if you cannot process face features, if you don't have neurons that are specifically processing face features, you're not going to be able to associate face features to the identity, right? So I think this is a bigger point where in order for something to be perceived, there has to be cells that are differentially responding to it. If I want to perceive color, there has to be cells that respond differently to different colors. Otherwise, I cannot perceive color. If I want to perceive faces and associate them with identities, I have to have cells that respond differently to different faces. If there is not a differential response to different faces in my brain, I'm not going to be able to recognize them. Does that make sense to everyone? And it's true of a lot of other things where you have to have a different response to different instances of that category in order to recognize different instances of that category. I guess sounds almost like a tautology, but the idea is that if I want to be able to process something, there has to be neurons actually representing those things. So when you look at this figure, it seems like we're assigning a job to every part in this system. And it would have been nice if brain actually worked like this, where you could imagine a car and there is a horn and there is seats and there is a part that is the tires and the this and that. So there are different parts to that system. There are different modules to that system, and they all work together to accomplish a function. As I said, that's a nice thing because you could take maybe in a cell phone, you could take the battery, replace it maybe, you could change the screen, or you could change the speakers of a computer. These are modules that you could take out and then fix them and you put them back in. Brain does have some degree of modularity, and you would know that because damage to specific areas seem to have more limited cognitive deficits. They're not broadly causing deficits. But there's a limit to that, especially in the brain. There's a limit to that, where all these areas are working together. They're densely connected. And taking out one area at the same time, impacts a variety of cognitive tasks. And at the same time, you could also compensate for that damage. So there's something about being connected, which is there's something about being connected that gives you, it gives you redundancy where you could perform a lot of different tasks through different pathways, or at the same time, you cannot really assign a single job to an entity. So these two go together. But I just want you to know that when you hear or read papers and they assign different jobs to different brain areas, just be, cautious of that, right? It's not like, oh, this area does this job and it doesn't do anything else, and no other area is doing that job. It's not that how it works. It's a network of connected areas which collectively are performing the task. This is the last slide I have here. And so this was left over from last week. I wanted to go through a few things and then we go into object recognition. One I wanted to show, again, I'm just trying to give you some more intuition of what the cortex looks like. This is the right hemisphere, right? You could all recognize it. This, we basically cut it, and then you're only looking at the right hemisphere. When you're looking at a pattern like this, which is a bigger like a big pattern, you actually should be able to see a ring forming, basically half a ring forming in the visual cortex. Why is that? What is this property? What is this property? Retinotopy, right? Because if it's a big pattern and then you're looking at a piece of light at light here and it's represented in a certain brain area, a bigger piece of light next to it is represented by an area next to it. So you should be able to see a half a ring, because this is the right hemisphere coming from the left side, for example. You should be able to see that ring kind of showing up in the visual cortex. If it was a letter A, you would see the left side of the visual field actually appearing on the right side. Now, this is a simplification in a sense that there's activity in the rest of the brain. It's just that you have to have, you could look at the fMRI, the functional magnetic resonance imaging signal that we talked about, before you have anything on the screen, and then after you have something on the screen. And then you can subtract them. And then it's not as simple as that, but let's go with that. Then you would see the ring appearing. That is the signal associated only with this visual input. But this is a very coarse signal. That's what we're talking about when we say fMRI is coarse. It's like a collection of 100,000 cells that you see as a voxel, as a piece here. In those 100,000 voxels, there are a lot of columns that we talked about, and they respond differentially, and some of them respond, some of them don't because of the orientation and all sorts of things. But on a coarse level, you should see something like this because of retinotopy. Because patterns are next to each other in the visual world, they activate tissue that is next to each other in the brain. Now, we saw something like this before, but in a different context. When I was talking about the somatosensory cortex, this is from a few weeks ago. And I said, well, if you maybe touch certain part of the hand, you'll see you see neural activity in certain areas. When you touch the knee, you see maybe activity in certain other area. If the face, you see it in some other area. There is generally, these are what we call topographic maps, meaning that areas that are close to each other in the body also are represented by tissues that are close to each other in the brain. So retinotopy is a form of topographic mapping. Like signals that are close to each other on the retina, which reflects the world. So these are things that are close to each other in the world, are represented by tissues that are close to each other in the visual cortex. But this also applies in a bigger context of what we call topographic maps. I just want you to know that these are similar. That's why, like features of the face would be next to each other here because, they're close to each other in the visual world, in the physical world. Okay. So another thing that you may see is that when you look, so this is a blown up version of the cortex. Blown up in a sense that while the cortex is folded like that, you have to put air in it metaphorically to blow it up. Well, after you blow it up, you can actually cut it open. And when you cut it open, you could get a flat surface, something like this. And you may see this in a paper. This is cortex, but it's cut open at this point. You basically, you put a scissor here, and then you cut it open that way. When you cut it open, right? So imagine the shape of the cortex is like this. And then when you scissor it up, you cut it open, it becomes like this. So when you look at one hemisphere, it looks like something like that, where the visual cortex is at the back, and then you go forward as you go deeper into the brain. So when I say V1 is just the patch of the surface, this is what it looks like. So when you cut it open, in a way, you're opening up the dinner napkin, and each area would be just a patch on that surface. That's V1. Next to it is V2, V4, and other areas. If it's a cortical area, then it would be a little patch on this surface. Does that make sense to everyone, how we got here? And if V1 has a topographic or retinotopic map of this space, these are from different animals. And this is a human. When V1 has a topographic map of this space, it means that in this, surface, different part of the space are represented, and the parts that are close to each other in the tissue. Okay, for example, here, I said this is V1. And when you look at it here, these lines tell you where V1 is, because these are the lines with V1. So you see the topographic mapping here, right? You see that these lighter blue areas are represented here. And then as you go in this direction in V1, you go in this direction in the space. This is just color coded to tell you which part of a space is coming from there. Now you could see that the yellow is down, or yellowish brown is down here, which is, again, because it's flipped. The yellowish brown is represented by this area, and then there is this spectrum going down. So this is what the retinotopic map on the V1 is. We talked about it, so I'm just reiterating some of the points so you get a sense of it. Any questions? Is this clear to everyone? Like, you guys have a better sense of the brain and how it looks like and what it looks like from different angles and those sorts of things now, hopefully. Okay. Time to go into object recognition. So a lot of the things that we talked about in this course going to build up to this lecture to understand what object recognition is and how it works in the brain. So object recognition is difficult for a few reasons, and we're going to just mention a few with examples. The main thing is that the stimulus that falls on the photoreceptors is ambiguous. So we have here a bunch of images that look very different from each other, right? There's like this picture and this picture and this one, and they look very different from each other. And when I mean they look very different from each other, it means that if you look at the pixel values, they're very different. If the pixel values are different and you look at something where the pixel values are different, the values that's going to be on the retina are going to be different. And that makes it very difficult. And you get an appreciation for how difficult this task is if you try to build a system like a artificial system that does object recognition. But we are very good at this. Like you could, if you didn't know what this category was, and you looked at these images, you can kind of form a category. You could look at all these images and be like, okay, I see what's similar among them, right? You could do some abstraction. Look at this and be like, oh, I couldn't see an abstraction. And then if I show you, and that's a category code, if I showed you which one of them belongs to the same category, you would say the left one. But the right one also has a lot of features that are similar to the left one, and it's very similar to the rest of this. It has similar sorts of color texture, or some facial features are similar. The background is similar. So when we look at some images, we're very good at abstracting what's really common among them and be able to recognize that object and learn a new category. If you didn't know this category, you look at these images and you learn a new category. And really, that's the challenge of object recognition, is that at what point, so if you look at this image down here, and you look at this image over here, do you think on the retina they have similar pattern of activation? No, it would be wildly different, right? Do you think in V1 they have a similar pattern of activation when you look at these two images? Yes or no? If I'm looking at this image for 2 seconds and you record from my V1, and then I look at this image for 2 seconds and you record from my V1, would those representations look similar? Why not?
Colors.
Okay. What's that? Different edges. The edge densities are so different between these two images. So V1 would be very different. Would V2 have similar representation? Should there be somewhere in my brain that these two images evoke the same representation or no? Same pattern of activation or not? So you think there should be somewhere in my brain that a brain area maybe has a similar pattern of activation. The neurons have a similar patterns of activation for these two images. Is that right? Why is that? Okay. So on a more abstract level, if I'm perceiving two things to be similar or the same, or have similar quality of some sort, then this has to be reflected in similar pattern of activation in my brain at some level, in some brain area. If I see two things that have the same color, it means that there is a similar pattern of activation in some brain area when I look at the two objects. So if there are two instances of the same category, and I look at these two instances, and I say, well, they're the same, it means that at some level in my brain, there's an area that gives similar activation for both of them. And I'm like, oh, they're the same. Does that make sense? So this doesn't happen in the retina. This doesn't happen in V1 or V2. It has to happen somewhere down the line. But that's the challenge of object recognition. How do you get similar representation for images that are so different from one another? In this case, you would say, on a pixel level, this image is very different from this image, for example, right? But on a more abstract level, they're very similar. And that's the challenge. And we want to find out where in the brain it happens and how it happens. Another challenge is that objects are often hidden or blurred in naturalistic environment. So occlusion is something we always deal with, right? We rarely see most of it for the whole object, right? But we don't have any problems with it. We know that there is a word here and it says cognitive psychology, most of it being occluded, scissors, or there's a pen parts of it visible, or pencil. So we deal with occlusion all the time. This is something that we're very good at. Again, just recognizing objects from a small parts. You could be driving on a rainy day and you have rain on the windshield, but you're able to see people and recognize them. Even you see a little bit of a person, and we're able to drive, and that has a lot of difficulty that we're good at. Again, a lot of this about how good we are at recognizing, and the reason it's important is that we forget, sometimes you take it for granted how good we are at performing certain tasks. Among millions of faces you've seen probably in your lifetime, you're able to pinpoint, for example, an individual face. Who is this? This is Saddam Hussein, right? And you haven't seen him for a long time, but you're still, who is this? Bush? Bush is a good guess. King Charles. This is Diana, and who doesn't know him? And Nixon. So you see, among all the faces you've seen, these are blurry faces of, some people that you may not have seen for a long time, or you've seen them at a different age, but you're able to recognize it. And that's extremely difficult for a model to do with such low grade, blurry information. Objects look very different from different viewpoints. When you look at this chair, it looks very different. And when I say very different, on a pixel level, it looks very different, right? You got to appreciate the problem that the brain has to solve here. You give it images from different angles that are very, very different. Like you look at an object from different angles, and they're very different from one another. But the brain has to say, no, these are the same object, despite the differences in pixel values or retinal image. Objects appear very differently in different illumination. So I have a demo for this. Because, let's see. So with all the things that I mentioned, right? Object, like categories could, appear ambiguous. The viewpoint could change, the illumination could change. They could be occluded in different ways. With faces, you could have facial features or not. How many different face identities do you think are in this image? How many people think it's 5? 4, 3, 2, 1. Not many people voting. So there are 2 identities here. So, but again, this is another thing that we have to deal with, is that, over years, you age and your facial feature change, and, the amount of hair you have changes. And also, lumination, luminance really changes, and, you have to find your angles, and the faces really change when you're lighting up from different angles. And again, this is a problem that the brain has to solve. When you see a friend and over the years you recognize them as different, as the same person, you're solving all these problems. The problem you're solving is that you have, across all these variations, you have to say, well, it's the same person. But you cannot be so invariant as to mix 2 identities. Does that make sense what the problem is? When we say you have to be invariant to something, When we say you have to be invariant to something, it means what? When a neuron is invariant to size, what does that mean? Or it responds the same way no matter what the size is, right? It could be active or not. If you're invariant to something, it means that variations in that thing does not cause variation in you. If you're indifferent to something in a way. A cell response could be invariant to size. It means that if you change the size, the response of this cell isn't changing. If it was a spiking, 10 spikes per second, changing the size is not going to change the number of a spike per second. But if I say the cell is coding or tuning or tuned for orientation, what does that mean? It's what? Yeah, so different orientation would cause different responses. So a cell could be invariant to a lot of things. It could be tuning for a lot of other things. So the problem of recognition is the problem of categorization or object perception is that you have to have representation that is invariant to changes within a category, but it's tuning information that could change from category to category, right? So if I want to recognize faces versus dogs, for example, human faces versus dogs in this case, I have to have a representation that is invariant to variations within face category, but changes when you go from face to dog category. Well, how would we do that? Well, let's put together everything we've learned so far. Let's say you have an image of a dog here, and I have V1 representation, primary visual cortex. And we said V1 mostly represents what? Orientations. And it does that in parallel for the whole image. What does that mean? That it does that in parallel for the whole image. Yeah. You want to add to that?
You're just gonna say like you're taking.
Yeah, so if I'm opening my eyes and I see the world and there's a dog in front of me, the image of the dog is reflecting on my retina. And that image gets processed, gets neural activity where there is maybe brighter colors, it's more activation, where there are, darker colors, less activation. And that image simultaneously for the whole object is getting projected into my V1. And there, all the edge information are being processed. So the neurons that have edges in their receptive field are going to be very active. And neurons that don't have any edges in their receptive field, they're not going to be active because they don't have the contrast that we talked about in their antagonistic center surround receptive field. But V1 has a representation of the whole object because every part of that object was on the retina. So every part of that object is in V1 and the representation is being processed at the same time. So V1 represents the whole object. Part of the object is represented much more strongly. Which part would that be? The foveal point, right? The point that I'm looking at gets a lot more representation than the rest of it, but the whole object is being represented. Represented, meaning that there are neural activity that correspond to different part of the object. Then these oriented bars, so there are cells that saying, okay, there is a bar here, there's a bar there, there's something here, there's something there. Their responses could get aggregated for recognizing maybe bigger bars, lines, or corners, or curvature. And then those activation could combine and become maybe shape. Those activation could combine and become maybe a facial part like eye or ear. And then those activations could combine and become a face. So this is what we call the feed-forward process of object recognition. And a lot of people believe that this is how object recognition happens, that you open your eye and when you look at something, when this dog was here and you were looking at it, the representation of the dog travels from your retina to V1, then V2, V4. V4 is called extrastriate because the back is a striate and this is extrastriate. And then you go to IT, inferotemporal cortex we talked about, posterior, central, anterior IT, and then that's how you get representation of the category. The representation, the spatial temporal pattern of activation that you have at the end of this pipeline tells you what that category is. Why do people think object recognition happens so quickly? I'm going to show you a demo of this. Which one is this? So, what are the instructions here? This is what we call a rapid serial visual presentation, or RSVP. And the paradigm is that you're quickly flashing objects around 100 milliseconds. And the subject's task is to recognize an individual object. Now, in this case, Buzz Lightyear is a character, right, from Toy Story. You all know what it looks like, right? I think it's a little robot. Is it that? It's an astronaut. Sorry, it's an astronaut. Okay, so when you see Buzz, just scream Buzz. Okay. All right, let's do this experiment. When you see Buzz, scream Buzz. Okay. Okay, yes. So I guess you're all able to do this task. And let's see, there was somewhere down here, right? Which one was it? Show. All right. Anyway, so you're all able to do this task, but imagine how fast this is flashing, right? For you to be able to recognize a complex object like that, when we have like 10 images per second, right? 10 images per second are flashing, and one of them is what you're looking for, and then you're able to recognize it. So when people saw this experiment, when they saw evidence that quickly, right, you could actually recognize an object, they were like, it should all be feed forward, right? Because when we talked about those axons, when we talked about, well, you have a neuron and then there's an axon and then you send the signal down the axon, the action potential down the axon, another action potential, another action potential. These are chemical processes and they take time. They take 5 to 10 milliseconds for each of these action potentials to travel down an axon. So for a signal from the eye to go to LGN, you have 5 to 10 milliseconds. From LGN to V1, you have 5 to 10 milliseconds. And then V1 to V2, V4, IT. So if you have 6, 7 stages or more, and each of them take 5 to 10 milliseconds at least, then it has to take between 60 to 100 milliseconds for the signal, for the first batch of these action potentials to reach the area IT, deeper into IT. Have a question?
Try to refresher on what forward is.
Feed forward, as it sounds, is activation feeding forward. So an alternative would be, in this context, would be that you have feedback signal, that not the information only moving in one direction, but also moving in the opposite direction.
Forward and turning back.
Well, if you have contextual information, that could feed back even regardless of what your input is. Or if you have an expectation, if you're cued for something, then you have like preparatory activation that could actually feed back even before you look at it. Well, I'll talk about an example there. But in this case, it's just that the activation is feeding forward. Feeding forward, I tried to explain this many times in a sense so you don't get confused, in that the idea is that you have a pattern of activation in the retina that causes a pattern of activation in LGN, right? It's not the same activation. That causes a pattern of activation in V1, that causes a pattern of activation in V2. The pattern of activation, well, how would it cause that? Well, a pattern of activation across many cells, these cells are connected to the cells in the next layer, and they release action potential, they release neurotransmitters based on their activations. And those neurotransmitters cause an activation in the next pattern. So this is the process, feeding forward.
Do you mean like a cascade effect?
Yeah. Yeah, feed forward is like a cascade effect. We talked about it. There are a few factors, right? When you have a pattern of activation in V1 that is caused by the image, the pattern of activation, you have many cells. These cells are connected to the next layer. Some of them are inhibitory, some of them are excitatory. And this pattern of activation, given those weights, the strength and the parity of those weights, releases certain neurotransmitters for the next layer and causes a certain activity in the next layer. But these activations, as we're talking about it, they actually compute something now. If you have two edges next to each other, and I'm going to go to a figure in the book just because so you see it. So this is up here are the earlier visuals, and it goes down, so the direction is different from there. So you could have in area V1, let's say this is V1, 2, 4, and IT. So in V1, there are cells that we're talking about that are detecting edges. If they're on the edge, if the receptive field is on the edge of the object, they're going to be very active because of their center surround antagonistic receptive field. If one part of their receptive field has activation and the other doesn't, they're going to be very active because they have contrast. So these two would be active, signaling that there are edges. Then if two edges next to each other are active, there is another cell. Let's say this neuron's receptive field is here, and this neuron's receptive field is here. And this neuron is connected to both of them. Where would be the receptive field of this neuron? It would be the sum of the receptive field of those two neurons, right? We talked about convergence. And because it's getting signal from these two, what would it respond to? Well, it would respond to this corner, because it would only respond if these two edges both are active and are excitatory, for example. If you have a bunch of corners, then maybe you get a shape. If you have a bunch of different shapes, you get an object. Does that make sense? So, and the receptive field of this cell would be what? The whole image. Yes, it would be the sum of everything that converges to it. Right? If you have multiple cameras and the cameras are looking and then are connected to each other, then at the end of it, you have the whole visual field. But why would these be connected this way? Well, because they're connected in all sorts of ways. It happens that this pathway within the bigger network is active for this instance. If the table moves a little bit, what would happen? Well, there would be another cell here that is detecting maybe vertical edge with this horizontal edge, and there's a cell that is connected to these two, so the corner would be signaled by another cell. Does that make sense? The space is tiled. So when you move or the object moves, the representation of that object kind of moves in V1. So different parts of this space you're getting signals about the edge from different neurons, but then because of convergence, it converges all to the same area in the higher visual. If you remember, I talked about this study. Well, you should remember this. I showed you a video of a study where they were trying to figure out which visual signal the V1 neurons respond, right? So what did they do? Well, they had lights. They would move the light. They would rotate the light. They would make the light bigger. They would change the shape of the light and record from a V1 neuron. First of all, they move around the light in order to see which part of the space is actually the receptive field of this cell is. And then in that area, they would just play around with the light. 60 years later, we're not that far ahead, honestly. Now, you could just put a monkey in front of the screen, and instead of an experimental playing around with the light, you could automatically generate the visual input. You're basically automatizing that process. How would you do that? Well, let's say I have a visual pattern, and I see how much the cell is active. I'm listening to the cell's activation. I show another visual pattern, and I see how much the cell is active. Another. Then I look at my visual patterns, and I see, well, this bunch of them really activated this cell. Let me just mutate them, combine them and see different elements of each of them. Then I show this image to the monkey and then see what the monkey responds, or this specific neuron response. Then I can combine them, right? Through this process of optimization, I could come up with stimuli that really drives this cell, that this cell goes crazy when you show it that stimuli. So in a way, this is a modernization of the experiment we saw before. Oh, not this. So you're looking at this process, how the image that is generated is changing as we evolve it. So this is an evolutionary type algorithm where you evolve the visual input until it really drives the cell. And you can see it arrives at something like this. This is what, as I said, drives the cell in the IT crazy, And you could see that, well, when you look at it, there are some visual features to it. You see some monkey face maybe, right? So when in early visual areas, we had a small receptive field, and you could characterize what derives the cell as like oriented bars. When you go higher up, it's not as easy anymore, right? Now you have complex patterns. You have a combination of a lot of different things that drive the cell. And I said, well, earlier studies tried faces and manipulating the faces and maybe line drawings and stuff like that. But you could automatize the process, and then you see, well, oh, this cell really likes these type of patterns. You could then record from another cell and see that, it likes this type of pattern. Now, these are not always interpretable, and we shouldn't expect them to be. These are complex cells that are signaling something to the rest of the brain or to their neighboring cells. So we shouldn't always expect them to have interpretable pattern of activation to complex stimuli. But this is, as I said, this is a modernization of the same approach where we are exploring the space of our visual stimuli, or it could be other type of a stimuli, in order to see which one of them maximally drives a cell. And we say, well, this cell likes this input, so that's what its job is. Its job is to detect monkey faces or something like that. But the reason you have that sort of selectivity is because of this hierarchy. Let's take a break until 7:05, and then we'll resume. Okay, so we talked about it. This level of specificity that you find, like or selectivity that you find in these higher level visual areas seem to support categorization, that earlier on you're detecting these edge information, but then as you go higher up, you're able to detect faces or objects or objectness in different ways. And detect here means that there are cells that respond to it, right? Similar things have been observed in deep neural networks that we build ourselves. So if you take a deep network, like we talked about a very simple artificial neural network, like 2 layers with a few connections, just to get some intuition. But there are object recognition networks that you interact with if you work with some of these chatbots that have image processing. You could feed it an image and ask them about the objects that are in the image. Or on your phone, there are a lot of tools like, AI branded tools to edit images, and they recognize different objects in the image, and you can remove them or inpaint them. When you look at those models, all of those models are hierarchical. They try to mimic the hierarchy of the ventral pathway when they are processing images. Now, a lot of them early on were hand-coded, like designed. That early area, they would detect edges, then later on they would combine edges to detect corners, and then shapes, and then objects. More recent ones are all trained end-to-end. So you have these connection weights or parameters between the layers. You input the image based on the pixel values of the image, and then across multiple levels, at the end, you want to classify the image. You say, okay, this is a dog, this is a cat. I'm not going to go into that. But what you observe is that in these networks also, this sort of hierarchy emerges. That when you look at, if you do similar experiments that we're doing with monkeys, now do them with a model. So you basically take the monkey out, in this case, take the monkey out, put a neural network in its place, and record from different units of that neural network from different layers, and see which patterns of visual input maximally drives each unit. You see something like this, that early layers are really these oriented bars. These are the center surround antagonistic stuff that we talked about, right? This is a on center off surround. This is an off center on surround. So you see similar units of selectivity, similar units with similar selectivity appear in artificial neural networks. And as you go up the hierarchy, you could see that later on, the input that maximally drives a unit looks like a dog face or a house or parts of a car or a taxi down here. So this hierarchy is a fundamental property of this system, where you have to slowly build up a representation that it becomes more and more selective to more complex patterns. Now, you could take this idea of selectivity to an extreme, right? And say, what if I have a cell or a few cells or a bunch of cells that only activate when I see my grandmother, right? That they don't activate any time else, only when I see my grandmother or I hear, the voice of my grandmother, or someone talks about her, or I think about her. Only in those cases I see those neurons get activated. And they call this the grandmother cell theory. And sometimes it's called the Jennifer Aniston neuron, and I'll tell you why. But the idea is that you are so specific, right? That you only activate when you see. So imagine this hierarchy has gone so far that you started from edges and not, and then you got to faces, and then faces become really specific for a specific faces. So you went all the way down to that level. Why is it called Jennifer Aniston cell? Because there was an experiment where they had these patients um, in the hospital, and they had electrodes in their temporal area, the area that we're talking about, which is the inferior temporal cortex. Why would they have electrodes in the temporal area? Yeah, so they're trying to monitor this patient, right? They have electrodes all over their brain and trying to continually monitor, see where the source of the seizure is. Source of the seizure is a pattern of activation that goes all in, all out. So if you look at a seizure response under when you see the brain activity of a seizure patient, is that the whole thing goes in and out. And so it's a very specific signature that you could detect. And they're being monitored, and while they're being monitored, they are being shown different images. In this case, they were shown images of Jennifer Aniston and other images of different people, of landmarks of Eiffel Tower and Golden Gate Bridge and other landmarks. And they saw that, again, there are two different ways we talked about it. There are different ways to show neural activity. One of them is a raster plot. So each row here is a trial, and you could see each dot here is an action potential. So for each trial you have this, you also could average this out. Say, okay, in this bin, how many action potential there are? In this bin, how many action potential there are? You could count them. Or you could show them up here as for each trial. So a lot of time when you show in the stimulus, you show it multiple trials and then you average out, you see, just to, get rid of the noise. What do you see here? Well, it looks like that this cell, among these inputs, this cell is responding strongly only when they see Jennifer Aniston in these different headshots, and they're not responding in other conditions. Like when there's a woman in the scene, or other cases, the figures are small, or when there are landmarks. Can we infer from this results that this cell is only active when they see when there's Jennifer Aniston being seen on the screen? No? Someone say no? Sure, right? So what are other options? What is it? Yeah, exactly, right? Maybe they're responding to the Friends TV show. How would you test that? Well, maybe you show people from that show, or you show the logo. So in a way, it's impossible to make that claim, right? Because there's always a category that you haven't tested. And in order for you to claim that this is so specific that only responds in that condition, you have to do, you have to try an infinite number of images. But the initial results seem convincing enough that, well, this cell is really specific. And it kind of, was taken as evidence for having some sort of cells that are specific to identities. However, as we said, like, there's no way, like, what is the other problem with this theory, right? Other than the fact that it's almost impossible to actually convince people to that extent, what is the other problem, you would think? What is the bigger problem?
There's no ultimate category. Like if you have to test infinite possibilities, that means there's not like isolated category for just one thing. Must be connected.
Yeah, but I'm thinking of a problem of what if it was true and we had a bunch of neurons for each concept or identity?
Yeah, I don't really think the strong thing but if there is like a sort of neuron that is specific to identity, like you're saying, it would be, wildly diverse, like from individual to individual. So like I would imagine if something like this did exist, it would probably be like some sort of like childhood memory, like a smell from my childhood would activate, some neuron to fire. But we'll all have different sort of experiences. So the sort of will be endless sort of, there'll be an infinite range of identities that would correlate to some neuron in the brain.
Yeah, that's a great point.
Kind of similar, like there's only like so much space in our brain, so
Yeah.
it would be kind of pretty wild to imagine that every single little thing has its own dedicated neuron somewhere.
Yeah, I think we'll run out of the space, like real estate, very quickly if we had a bunch of neurons dedicated to a concept, right? Like there's so many faces you've seen in your life, so many on so many TV shows or in your life, and there are so many other concepts that exist in the world that you lived with and you experienced with. So it's really not efficient for a system to dedicate a little piece of the space to each single one of them. Imagine if you had a phone book and you color coded them, right? Like for every person, you had a color. You would run out of color very quickly, right? So it's not really this sorts of specificity coding is not efficient. This isn't a schematic of that. So you're saying, okay, I have 3 friends and this neuron 1 is only active for Bill, 2 for Samantha, and 3 for Roger, right? So what is the problem with that? Well, what if I lose this neuron? Then I would lose my memory of Samantha, or I would not identify Samantha anymore, right? That's a problem. The other problem, so what happens if I ran out of Samantha, like neurons? So can I not learn new faces? So what is the alternative? The alternative is that instead of me assigning a neuron to each face would be represented by a combination of neurons. Okay, so this is what we call its distributed code. Now, if I want to add another person to my memory, how would I represent that in this space? Let's say I make a new friend, and I want to represent that. What is an example way of representing that? So what is an example pattern? Right? I could have, what's that say? Yeah, low on all 3 of them, for example. If I have a pattern that is low on 1, 2, and 3, it does not overlap with any of these patterns, but it's a separate pattern. And a downstream neuron, a neuron that is listening to all 3 of them, would know that it's different from these 3. So if there is a neuron there that is taking an average of all 3 of these activations, then it would be a different pattern for this versus this new pattern, right? So go with the phone book example. If I have 1 color for every friend, right? I ran out of friends, maybe at 12. But if I use a combination of colors for every person, like one person, is green and a little red and a lot yellow. Another person is, a little yellow, a lot red, right? If I use a combination of those things and use that to represent every person, then I'll be representing a lot more people, right? Does that make sense to everyone? When we have this distributed code, then it's much more efficient because I can now represent a lot of different things. Another thing is much more robust. If I'm representing a concept by an activation of millions of neurons, then what's a few neurons? right? If I lose a few neurons, I'm not gonna lose that memory, or I'm not gonna lose that percept, right? And neurons die all the time. So it's important for a system to have two important properties here. It has to be efficient in a sense that it could represent a lot of different things with the same amount of neurons. And also it has to be robust in a sense that it doesn't suddenly fail when you lose a few neurons. So that's the property of distributed code versus a specificity code. And we mentioned that you could represent different people with different patterns of activation. And you can imagine a downstream neuron, and I say that meaning that, well, these are all at a certain level. There are other neurons that are listening to these neurons. And their activity could change depending on what kind of pattern of input they're getting. Okay, so representing the patterns of activity distributed over many neurons, we call that distributed coding. You see this a lot in life. As I said, we do use these sorts of distributed coding for a lot of things. It's more powerful in a sense that you could represent a lot more different patterns with the same amount of bits of information that you could actually play around with. And also you have graceful degradation, meaning that losing a few neurons is not going to mean that everything's lost. Questions about this?
Can I ask you a question about like this idea of concept or categories?
Sure.
So like I'm just having like a little trouble of like, so we're talking about these like neurons and neural pathways that can detect edges and light, etc., like sort of qualities of objects to differentiate them in like homogeneous space. But like how do we get from there to like a category of something? Like, I mean, perhaps like maybe some categories are like pre-programmed, like recognition of faces, maybe, and like I can see that being argued from an evolutionary perspective. But then things like koala or table, like those can't be pre-programmed. That they must sort of come from like experience, from like from the world of sense. So yeah, like how do we? It's like are concepts like learned and then stored like in the brain? Is that the proper language, or how do sort of neuroscientists conceive of that?
I think so. There are different ways to think about that. So there's something about naming objects. So which is, let's say it's a human thing, and let's say we set that aside. Let's think about an animal. An animal can categorize this stuff, right? A dog can know that, well, this is my owner or not, or that this is the food that I eat or not, right? So
Yeah.
they form categories. They have categories probably for chairs that they sit on and tables that they could jump on and those sorts of things, right? How do they form those categories? So none of it is innate, right? Because we didn't grow up in a world of these objects, but meaning that we didn't evolve, and these are all very new objects. They're all learned. How are they learned? Well, they're learned in a sense that we were talking about here. Let's say the table, right? How is it learned? Well, there were a lot of common machineries in terms of detecting edges, let's say. And those machineries are able to detect combination of those things. And then as you go up the hierarchy, you would have a spatio-temporal pattern of activation that you know it's a table. You could name that activation, that pattern of activation, but you don't have to, right? If you see a chair, like a dog, when they see a chair, they could try to sit on it, or a sofa, they try to sit on it, right? They act upon it. But you get to that category through this hierarchy. So what does it mean for you to recognize it, for a dog to recognize a chair? It means that when they see a chair, when they see another chair or another chair, there is a pattern of activation in IT that forms that is the same across all of those. Does that make sense? Through this hierarchy. Because you're detecting the edges come together, they become a shape, and then those shapes combine together and they become activation. But there is something common among all those chairs in that activation. Does that make sense? Does it
Yeah,
answer your
but-
question?
No, definitely, it's very helpful. And then like, just like really briefly, it's like interesting with like chairs, for example, like, yeah, there's the sort of more arbitrary part of like naming them, but then there's also like, there's like, that's a chair and that's a chair, and these are like, there are all sorts of different chairs, and then I can go see a chair that I haven't ever seen before and still sort of subsume that under the category
Mhm.
of chair. And like, even if you take the naming part away, like I could see animals like that I've never seen before. And like, just sort of by recognize them as like part of a group, and that group sort of being meaningful, independent of my perception.
Yeah.
Yeah,
So that's the difficulty, right? In a sense that you're learning multiple samples and then you're generalizing to a new one.
Yeah.
So let's say, how does that happen? Well, there has to be some commonalities. If I'm learning multiple categories, I'm kind of learning what's common among them. And that commonality is encoded in these connections,
okay.
right? So yeah, so if I'm looking at a chair and I extract certain features of the chair on lower level, they activate some other, eventually they all have to lead to these spatial, like a distributed representation of a chair.
So you're saying basically like, by seeing different kind of chairs, like it's like training the neural network.
Yeah. So when you see if it was in an artificial network, the way you train it to recognize a chair or anything else is that you have to show it a lot of examples of the chair and say, well, at the last level, give me similar representation for all of them. The units that are at the last level, they give you the same representation for all these instances of a chair. Then when you go to another thing, let's say dog or table, the representation, whatever pattern of activity should look different from the chair, but for all these tables should look the same. Does that make sense? So what are you learning? You're learning to be invariant to changes in this category, but the pattern has to change when you go from one category to another. So we're learning two things at the same time. We're learning to vary, the representation has to vary when you go from one category, chairs to tables, but when you go from one table to another table, the representation should stay the same. And all of that is encoded in the weights of this network.
So basically we are like, developing the world of ideas by experience.
Sure. For everything has to be through experience, yes.
Well, the world idea is not basically Plato, like,
I'm not familiar with the,
like, no, oh, never mind.
yeah. So you could bring these ideas together, the idea of distributed coding and specific coding, because fundamentally there has to be some specific coding. And that is specific coding because after all, you can recognize your grandmother, you could recognize a specific person's face or a specific thing that you saw somewhere. So there has to be a specificity here, but it doesn't have to be at the concept level. It can be at the level that you build those building blocks of a concept, right? You could have a certain type of, in this example, you could have a certain type of hair or a facial structure, or, you know, certain that each of them is represented in a specific distributed way, and then together they represent a face. Does that make sense? So it doesn't have to be that there are a few cells that always respond to your grandmother, but it could be that there are different population of cells that are responding to lips, and they represent the lips, and depending on their pattern, they have certain kind of lips, and then another one, certain kind of hair. And then when they're all active together, collectively in a distributed way, they represent a person. That's the idea. Okay, so far we talked about this process of feed-forward processing, that you have an image coming in, and the image gets represented in early visual areas, and it goes along the hierarchy, and then it eventually gives you a pattern of activation depending on your experience that corresponds to a category. And for a lot of animals, that category only means an action, right? If it's a prey or a predator, you act differently because you recognize it. You see, okay, I'm seeing a predator, I should run away. So if you go along the hierarchy, eventually you have a pattern of activation that leads to an action. But for us, it becomes a lot more complicated because the language is involved in it and everything. So there is a lot more to recognition, right? We don't always just open our eyes and recognize things in front of us. We could lose a lot of context. We could use a lot of things that we know about the physics of the world, of light, of all different stuff to recognize things. For example, when you look at an image like this, you get a different perception. A lot of people would get a 3D perception. Like when you look at this disc, does it look like it's jutting out or does it look like it's going in? It's coming out, right? But how about this one? In. So this is on a 2D surface, right? For it to give you a 3D perception, I think it relies on our knowledge of, the physics of the world. We assume and experience that light always pretty much comes from the top. So if light comes from the top, for you to see the top of an object lighter, it means that the top has to be out, jutting out, like this. It has to be jutting out for you to see the top lighter. And in order to see the bottom lighter, it means that it has to be going in. It has to be indented for you to see the bottom of the objects be lit up. So knowing about the physics of the world or having experienced the physics of the world gives you information about, changes your perception. When you look at these sand patterns, does it look like, do these areas look like they're going in or coming out? So what if I show you this? These are coming out, right? Compared to these, what do we have? Do people see this at the indentation? And these kind of coming out, amounts? So it's the same picture that is turned upside down. But because, right, we have an assumption that the light usually comes, in the northern hemisphere, you kind of assume that the light is somewhere out here. And if the light is somewhere out here, for you to this part kind of light and this part kind of dark, it means that this has to be going in, like this has to be an indentation. But in this case, for this part to be gray and this part to be light, and the light coming from this part, it has to be a mount. So you the perception of the object, well, it's the same image, but because we have assumptions about the physics of the world, then we'll perceive it in a certain way. So it's a heuristic, but also it's kind of wired into our perception, right? You can't really help it a lot of times to perceive things in a certain way because our experience of the world has been always agreeing with this physics of the world. Another thing that really impacts our perception is scene context. So what is this object? What? So a plate of food, you could say. What is this object? Car. So why would you think this is a car? Well, this is a very blurry image, right? So you're relying on something else to detect this as a car. And you're kind of, maybe that's a building and there's a road, and what else could it be? It has to be a car. And here you're kind of recognizing the people, a table, a bottle here, a glass here. So this has to be a plate of food. But in reality, these are the same patterns that are copied over, right? So this is the same pattern here to this one. It just if you take out the rest of the thing, you really cannot recognize anything. It's just the contrast is different to match the contrast of the image. But it's the same pattern. And it's copied over. But when you get a scene context, you rely on that scene context. You're like, okay, if I have to recognize something and looking at it, it's not giving me enough information, then I'll rely on the context. And we do that all the time, right? We use the context that the object is in or other information that we have in order to recognize the object. Okay, so there were some, I'm going to talk about this experiment. There has been some experiments to understand what the scene context effect is. So in this experiment, people were shown images of the kitchen, for example, and then there were three different examples to see. So the experiment was very simple. You have an image appearing, and then you have another object flashing. It could be a slice of bread or, white bread. It could be a mailbox, or it could be another object. In this case, because of the cheese and the knife and other stuff, the toaster, the target, scene context target would be bread. The scene inconsistent target, but visually similar, would be this mailbox. And then the scene inconsistent target, which is not visually similar, then it would be this object, whatever it is. So the goal here was to see which one of these objects would be better recognized. So this is a paradigm that you could use, where you could show an image and then you could flash the second image for, I think, as short as like 20 to maybe 80 milliseconds. So it's somewhere really fast. And you could see how many people are able to recognize it and what is the effect that the scene has on that recognition. What they found out in this experiment is that people were much better at recognizing scene consistent objects. So if they saw a scene of kitchen and they were shown these objects flash, they were much better at recognizing a briefly flashed bread versus mailbox. The target consistent with the scene context was identified more often than the inconsistent object. So this kind of shows you that when you are under constraints in terms of the duration of the time that you have to look at an object or the type of information that you can get from the object, then you're likely to rely on the context of the scene, right? So in this case was another example of this. Because the objects themselves were blurry, you rely on the scene context in order to recognize them. In this case, because the objects are flashed, so you can imagine if the second object was not flashed, if it was actually there for a long period of time, or beyond, let's say, 120 milliseconds, you would not, you would not get this pattern of activation, the pattern of behavior, right? You only get this because people don't have enough time to gather enough information from the object itself. Yeah.
Would they flash the inconsistent targets, but people just not able to say what they saw, or are they not like
in this experiment, people would know that something was flashed. And it was an open-ended question, meaning that you show a scene, you flash an object for a very brief period of time, and then you ask people to write down. And in these experiments, usually they encourage the participant to guess as much as they can, right? Like people were like, oh, I didn't recognize it. I saw something flash, but I didn't recognize it. But you encourage people to write down whatever they thought they think they saw. And you just see how often they get it right. So a lot of experiments, you have to ask people to guess because they're not confident. Yeah.
When it says visually similar, is that talking about to the scene or to the
To the loaf of bread. Because you want to, if you just you know, submit a paper and then you only tried the loaf of bread and another object, a reviewer would say, well, but you didn't try something that was visually similar. So how do I know if the loaf of bread, it was because it was consistent and not because of its shape or something like that, you know? So in order for preemptively addressing that issue, you would try to find this control where you have visually similar to your target, because the effect is really here. And then you have something that is like even visually dissimilar and inconsistent.
Whether the visually dissimilar object was observed more than the visually similar one, because it seems like because it's novel, it might stand out.
Novel in a sense that is inconsistent.
Novel in the sense that between the three objects flashing, it's the only one of that like shape. So I was just curious if
You mean this one, inconsistent one?
right, but they're both inconsistent, both B and C. I was just curious if C is more likely to be remembered.
Oh, I see.
Or guessed.
I'm not sure. You know, I was looking at this paper earlier.
Or it's kind of a like off topic, no worries.
I was actually looking at the paper. And the objects are actually a little different. As you can see, this was an older study, so they cleaned it up in that picture. It's from the 70s. Yep, you could check out the paper. Okay, so the idea is that a lot of time we rely on context to recognize objects. When we don't have enough time or the information coming from the objects are ambiguous because it's blurry or maybe it's occluded, and for all the reasons that we talked about that makes object recognition difficult. So you could have a system where you're actually taking top-down facilitation into effect. So a simple idea would be that you have an input that is processed by early visual areas, and this goes through the ventral visual stream. This is the stream that we talked about in the past couple of weeks, where you go from the early visual area to the inferior temporal cortex. So fusiform gyrus, that's like fusiform face area, is in this, it's down here. It's based, you can think of it as the infratemporal cortex. That's the area. And this pathway is really fast. And we saw an example of it when I was flashing images at a really high rate and we were supposed to detect an object. We were able to do that. So it shows you that this pathway is quickly, every 100 milliseconds is being, processing an image. Like basically every 100 milliseconds an image enters and exits basically. But you could have a much slower pathway where the early visual area response goes into the frontal area of the brain. And from there, you have these top-down modulations coming to modulate the response of the IT. So in the case here, for example, all the reasoning about what the scene is can later on impact your recognition. It's a slower response and it goes through probably the frontal areas and it gets, processed. And then from there, it could come down and modulate your response. Top down, bottom up are loaded terms and we'll encounter them a lot more when we talk about attention. In this context, bottom up and feed forward are almost identical. So you could talk about bottom up input like it's the feed forward input. Top down could be local Like you could have top-down input from, let's say, IT to V4 and from V2 to V1. But in this case, top-down means that it's coming from the frontal area. So you actually have reasoned about the image, and those reasoning has impacted your processing. As I said, we'll talk about this. What's top-down, what's bottom-up? Again, we were talking about this top-down facilitation. What's top-down and what's bottom-up? It's not easy to understand, and people don't agree on it. For example, you could say, is this a top-down or bottom-up? I don't think anyone would agree on. Like people wouldn't agree on it. Like some people would say, well, this is a top-down effect where your physics knowledge impacts your perception. Some people, well, no, you have had exposure to the world as it is, and that impacts your bottom-up processing. So, so we'll use those terms, as I said, when we talk about attention more, but know that they are not everyone agrees on what's top-down, what's bottom. But in this case, in the case of recognition, this fast feed-forward processing that we talked about here is what we call bottom-up. That's what that means. So you're coming from early visual areas and going up the hierarchy. So bottom-up or feed-forward. Top-down would be, well, if I had, so in this case, for example, these people put, they said, okay, again, this is an emergent understanding, where people are putting feature processing in area IT, that you process the features of the image and categorical features as well. But the actual categorization or identification happens in prefrontal cortex. So they assign that job to prefrontal cortex. Now, what's the difference between categorization and identification? Categorization is that you're saying, well, these are all faces and these are all dogs. But identification is more of, oh, this is my uncle and this is my dog. So it's even more specific than categorization. Categorization is broader. But both of them involve a lot of, even when you want to recognize your own dog or your uncle, you have to have a lot of invariances because your uncle going to look different every day, right? They could have facial hair or not. They could be wearing something else. They could get a haircut, and they change over time. You may not have seen them for a year or so. So you have to, for every act of categorization or identification, you have to have invariant response that are not serving you. You have to be invariant to it. So changes in a person, when you want to identify them, you have to be invariant to those changes. But if you go from one person to another person, then those changes have to be different. You have to change to it. But if you wanted to recognize a human, then you wouldn't care about those changes. Does that make sense? So categorization in this case is hierarchical, that there are hierarchies to them. Depending on what your goal is, the response have to be invariant to some changes, and it has to vary with some other changes. Does that make sense? Yeah.
Is it saying that if the image is like clear and to identify, then there's just this quick processing from the early visual stars, but if it's like low spatial frequency, almost difficult to identify, then the top-down versus?
Not exactly. This is saying that the top-down information does not rely on high-resolution information. So it's saying that, again, this is a schematic. But if you had a visual input, the top-down could be on low spatial frequencies. Low spatial frequency means that it's Billiard, right? So the image, you could have low spatial frequencies, and then based on that low spatial frequency image, get the top-down information. It doesn't have to be, because you don't really have high-resolution visual processing outside of this pathway. Does that make sense? So this pathway only does high resolution visual processing. If you're going through other pathways in the brain, you don't have access to higher spatial resolution, but it doesn't matter. You don't need higher spatial resolution. For example, in this case, You don't need higher spatial resolution to decide that, okay, this is a road and I want to recognize this as a car. Even the lowest spatial resolution will do. That's the point.
Is that like evidence against the grandmother?
It's evidence against the extreme version of grandmother cell. So the extreme version of it, where you have a bunch of cells dedicated to a concept, yes. So we're saying that that's a weak theory for a couple of reasons. The reasons being it's not robust, that if you lose some cells, you lose a memory, and it's not efficient in that you run out of a space. But there's a middle ground where we could think of having a specific feature that together, collectively, they could represent something more specific, right? So you could have, as I was saying, if you have a combination of color intensities to specify a friend, right? You don't have one color to specify a friend, but a combination of color intensities or could specify a friend. So you could have a distributed code that are themselves specific and collectively represent a specific object. Yeah?